Hi friends, welcome back to the JK Biology, and today I will be describing about the RNA splicing. Before this lecture I have described about the polyadenylations and the 5G cappings. Okay, so the RNA splicing is the most important topics. That's how it takes place, and their transcription reactions, their different factors as like spliceosomes, that's complexes involved, I will be described. So the first description I will be say about the transcription reaction. That is the most important reactions in the RNA splicing. So, okay, you know that in the eukaryotes, RNA, pre-mRNA contains introns as well as exons, but there is no need of introns, so only one coding sequence is the exons, and exons is the required that make the proteins. Okay, so the removal of the introns is required, and then how it process takes place you can see here. That suppose that there is the mRNA 5 and RNA splicing occurs between bases, that is the conserved sequences in the eukaryotes, GU, 5GU and AG. Okay, so there is the GU sequences that is conserved, that is 5 to 3. Okay, this is 5 to 3, and this is also called 5 splice site. Splice sites, point out, this is called 5 splice sites. Okay, this is the pre-mRNA. I will showing you the structure of pre-mRNA, that is exon. Okay, L1, suppose that I am naming is exon L1. Okay, and then here is the intron, there is the adenine sequences. In the intron, adenine sequences, that is the most important. I will be described about it. This is present in the intron. Okay, within the introns, not at the end of intron. Then again AZ. This is naming in the blacks because this is also present in the intron, and this is also I am showing you blacks because this is present in the intron, GUAZ. Okay, and here is the L2, 3 exons. This is pre-mRNA sequences. This is total pre-mRNA sequences. Pre-mRNA. Okay. Then there is need to removal of these introns. Removal of these introns. So how this occurs? There is the more complexes. First I will say about the transcription reactions, then I will be described about further the complexes that is involved in the spliceosome are making loops. These problems would be solved by making loops within the sequences. So there is adenine where OH, 2 OH of the adenines. If you have imagine in your minds, draw the figure of adenine, there in the 2 position of the carbon, there is hydroxyl groups in the adenine, and these 2 positions, points are, this is the important 2 position of the OH react. Okay, this is another react here, react here, where there is the junction of exon first and intron, that is OH, but where it react there is the phosphate, the OH of the adenine within the intron react with the junctions of exon first and GU of the introns of the phosphate. 5 phosphate, 5 splice sites. Point out also this is the 5 splice sites. So there is 5, that is 3. So these OH, 2 OH of the 5 splice sites react with the phosphates. This may be also questions. This may be also questions. 2 OH of the adenine react with the 5 splice sites. 5 splice sites of the exon intron junction. This is called the first trans-esterification reactions. This is called the first trans-esterification reactions. Okay. If you asked by questions that first what is the process or what happened in the first esterification reactions, then the answer would be 2 of the adenines and 5 splice sites of the phosphate junction of the intron and exon is called the first trans-esterification reactions. Then what would be the happened after first trans-esterification reactions? You can see here first trans-esterification. Points out again, this cannot alone take trans-esterification reactions. There is more complexes. I will be described that there are some U1 factors, U2F factors, and many U2, U4, U5, U6. This is the spliceosome complexes that is processed in the causal bodies of the nucleus, and so these complexes involve to making the lariat. So that they goes to the proximity of the adenines. After formatting lariat, these phosphate goes to this direction, so the trans-esterification reaction would be occur, and the transfer of the ester bonds would be takes place. Okay, because the close proximity. So there is the many splicing factors. So I will be described about the side, but first I will be described about the trans-esterification, because this is also most important. So you will see here that there is the first trans-esterification reaction. So what happens? This is the breakage, that is L1,5, and remaining of the OH breakage, and these make bonds with the phosphate. These make the bonds with the phosphate. So imagine what would be the happens. These bonds here. So there would be the structures plus AG A here G. Okay, these would be the structures L2 and 5. Points out, this would be the structure that react with phosphates, then form the lariat. It is becomes as like lariat. Okay, so also this is the L2, that is the exon required to bind with this, to join with this. So there is also phosphate. Okay, there is also phosphate, and there is free hydroxyl group of the L1 react at once with the phosphate of exon 2 and intron junctions. This is the AZ and L2 introns and exon junctions. Then it will react with the phosphate of exon and intron junctions. So again there is, if you know the mechanism of the reactions, by the transferring of the electrophilic or nucleophilic attack, then one breakage of the bonds and one formation of the bonds. So there is breakage of the bonds. Okay, there is also bonds. I am showing you there is a phosphate, there is bonds, so this will break, and additions of this phosphate to the that of OH. So what would be the happens? Again imagine these exons becomes joined, okay, and this becomes removed. So this is called the second trans-esterification reactions. So again questions, what would be the happens in the second trans-esterification reactions? Then you can say that exons first of the OH hydroxyl group react with the intron exon junction of the phosphate and making what? L1, 5 and L2. Okay, 3. There was, sorry, this was the 3 and then 3. This is exon plus this is intron. Sorry, AG, A and G. This becomes intron. So always introns is released in the form of lariat, and in the further process it will be degraded in the nucleus. Okay, and this is the exons that make the protein after making the proteins. So this was the trans-esterification reactions. Okay, this was the trans-esterification reactions, that is also 5 to 3. Okay, again I am describing about the complexes that is involved. First, where points out, keeping in mind that this is the process of trans-esterification reactions, and in this process spliceosomes complexes involved that form the lariat, so trans-esterification reaction occurs. Okay, because in the at the time of formation of the lariat by the spliceosome complexes, the trans-esterification reaction would be occur, but I am describing separately because you can understand easily the trans-esterification reactions. Okay, so there is the pre-mRNA, where first U1 is the snRNA, snpRNA, you can say small ribonuclear proteins that is 200 nucleotides long, is involved in the formation of the spliceosome complexes. So the all U1, U2, U4, 5, 6, all these are snpRNAs. Okay, so U1 first binds to the 5 splice sites. This is also, that this is the 5 splice sites. Okay, 5 splice sites, and this is BBP at protein bind to the branch. This is called the branch points within the introns, that is adenine, and adenine becomes nearly about 45 and maybe 20 nucleotides long within this introns. Okay, and so also there is U2AF factors that is also binds within the introns. Okay, that is also binds within the introns. So spliceosome complexes involve ATP. It's require ATP, but trans-esterification reactions does not involve or does not require ATP. Okay, so point out, this is the U1. When there is another, this is called the E complex. What is the E complex? BBP, U2AF in the introns, and the branching points BBP proteins binds and the U1 snRNP binds, then this is called the E complexes. Okay, and then after U2 replace the BBP at the branching A sites. Okay, AG, A, GU, L1, 5 and this is U1. Okay, and here U2 replace this U2AF and that is L2. Okay, this is replaced, U2 replace the BBP proteins, U2 replace, points out, U2 replace. When the U2 binds to the branching sites, there is formations of the close, that's become GU goes to the proximity. This is U1, this is U2, goes to the proximity, okay, and further there is U4 by U6 and U5. This is the complexes again comes. When these complexes comes, there is removal of U2AF factors. U2AF is released. Okay, this is U2AF is released, and this also I am saying you, this is the complexes A complex. This is A complex. You can see here that what is the combination of the RNAP ribonucleotide proteins of the A complexes. You can see here. Okay. After then removal of U2AF, U4, U6, U5 will comes, and again the structure look like L1, 5, GU, U1. That's formed in the form of lariat. Sorry, A, AG, and L2, 3. So there is GU. Okay, U1, U2, and all these U4, U6, U5, U4, 6, U5. These are the complexes. Okay, these are complexes, and that is called the B1 complex. Points out, this is called B1 complexes. What is the combinations of the B1 complex? U1, U5, U4, Y6 and U2. Okay, then first release U1. First release U1 and 5L1 AG A and L2, 3 sides. Okay, this is also I am showing you for simple examination. You can see on the basis of the colors. You can see on the basis of colors, not differ from that is black, that is blue. You can see L1 is the exams and also L2 is the exams. Okay, so after then again release of the U4, release of the U4 ribonucleoproteins, and then this is called okay B2 complexes. B2 complexes, this is called the B2 complexes after removal of the U1s from the B1 complexes. This is the B2 complexes. Okay, then U4 is released, the structure would be formed, that is called the C complexes. C complexes, that is called C complexes. So this is the sequence: E complex, A complex, B1 complex, B2 complex and the C complex, and the structure would be formed, that's in the form of the lariat. The intron is removed as I have described before this. After then what structure would be the released, that is 5 L1 L2 3. This is the exams forms, and in the form of lariat A AG, and attaching after removal of the U4, you know that's U4, 5 and 6, U4, U5 and U6 remains with the introns, and this introns is removed and goes to the degradations, but the exams is remains for formation of the proteins and further process. But you know that's these ribonuclear proteins becomes recycled, it does not degrade it. it's become recycled, but the introns become degraded. So this was the complexes that's involved in the trans-esterification reactions and also involved in the RNA splicing. So this is the most important topics I have described. Okay, and further in the lectures I will be described about the rRNA processing, tRNA processing in the eukaryotes. Okay, and so you know that after removal only exons is remained, that is the coding sequences. So after formation of the cDNA from the mRNA, you know that after splicing complete mRNA, matured mRNA is formed that no contains any introns. If the from the mRNA we make the cDNA, that cDNA does not contain any introns. So this cDNA becomes the intron free, you can say. And so if you want to interested to know about the cDNA, then you can go back to the lecture of the cDNA library formations in the JK biology, and you can easily find that how process of the cDNA formation takes place. Okay, so cDNA is the intron free, and also matured mRNA after processing, after splicing, it's become intron free. Okay, and also in the prokaryotes the mRNA becomes without introns, so it will only contain the coding sequences. Okay, so hope this lecture would be very beneficial for you. If you like this lecture, then please click on subscribe button and also share for your friends. Thank you.